This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 1121, recorded on June 7, 2024. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Many of you know, because you've written to us, that an opinion piece was published in the New York Times in the past week by Alina Chan, and the title is Why the Pandemic Probably Started in a Lab in 5 Key Points. We've received many email requests to discuss this, and so that is what we are going to do because the title isn't correct. And we're going to go through each of the 5 key points and tell you why. But
and I think before we launch into that discussion specifically, Jolene, do you want to run down the, this is not the first time this has come up on the show?
Yeah, so we're going to go through and discuss some of the article, but we have done this in much greater depth, actually discussing the papers that address these points, as well as talking to some of the scientists and just to remind people of where they can hear about this discussed in greater depth and kind of listen to the evidence that has been presented. I wanted to run through a few of the episodes that we have. We'll put a list in the show notes that where these things have been discussed. So TWiV 1019, Eddie Holmes on SARS-CoV-2 origins. TWiV 940, also Eddie Holmes in on viral origins. We have TWiV 861, rough draft of Omicron origins. TWiV 762, SARS-CoV-2 origins with Robert Gary, where we did talk about things like the receptor binding domain and the furin cleavage site and the 2 lineages circulating in the Wuhan wildlife markets. TWiV 760, SARS-CoV-2 origins with Peter Daszak, Thea Coulson, Fisher, and Marion Koopmans. And TWiV 777, SARS-CoV-2 fitness with Ron Fouchier, fitness and the ability of SARS-CoV-2 to replicate and spread in other organisms. There is a long list. I'm not going to read all of them, but look in the show notes for many more instances of TWiV episodes where we discussed with guests and just discussing papers a lot of these points we're about to go through.
And if you're on YouTube, that'll be down right below this video.
Okay. So there are 5 points in this opinion, but there is an introduction. And let me read from the introduction. Quote, a growing volume of evidence gleaned from public records released under the Freedom of Information Act, digital sleuthing through online databases, scientific papers analyzing the virus and its spread, and leaks from within the U.S. government suggest that the pandemic most likely occurred because a virus escaped from a research lab in Wuhan, China, end quote.
Ooh, secret newly revealed knowledge. Woo!
In fact, most evidence that has been published in journals, which we have covered in many of those episodes that Jolene talked about, supports a natural origin, not a lab origin. And Dr. Chan is ignoring most of those data because they don't fit with her narrative. And we understand now, and we've talked about this a lot, the pandemic began when a virus jumped multiple times from animals on sale in the Huanan seafood market in Wuhan to humans. There are several papers on this, and they basically are devastating to the lab origin hypothesis. They show that the market was the early epicenter, that 2 lineages were circulating there, that susceptible mammals like raccoon dogs and civet cats were sold in the southwest corner of the market, and we have environmental samples from there that showed the presence of both animal DNA and viral RNA from SARS-CoV-2. So this statement, a growing volume of evidence suggests it occurred because a virus escaped the lab, is simply wrong.
If people are particularly interested in the things that Vincent mentioned, I particularly found the discussions in episodes 876 and 995 to really be particularly enlightening, and the data that we saw in those two really was very clarifying to me and made it very obvious to me.
Yes.
So
Yeah, and I just want to harp a little bit on this two lineages thing, because when I saw that, I mean, when those data came out, um, it's a subtle but really damning point if you want to say that this came from a lab, that there were two genetically distinct lineages of this virus at the very, very earliest days of the outbreak in Wuhan, and there is simply no way to get to that from a leak from a lab on a virus somebody was working on.
Yeah. All right. Point number one. Quote, the SARS-like virus that caused the pandemic emerged in Wuhan, the city where the world's foremost research lab for SARS-like viruses is located. Bats in other parts of China have not been found to carry viruses that are as closely related to SARS-CoV-2. All right. So we have to talk a little bit about what is closely related to SARS-CoV-2. The closest relative at the onset of the pandemic is a virus, is a genome sequence called RATG13, which was taken from a bat in a cave called Tongguan Cave in southern Yunnan, which is about 1,800 kilometers from Wuhan. RATG13 is 1,200 bases different from SARS-CoV-2. That's 96.2% similarity. So RATG13 could not have been the progenitor of SARS-CoV-2.
And 96.2% genome identity, we were talking a little bit about this before the show. You know, if you get 96.2% on a test, you aced it. But if your genome is 96.2% identical to something else, that's, what is that, humans and sooty manga bays or something? I mean, that's...
yeah, something like that. And this is particularly important because coronaviruses for RNA viruses are quite large. So the genome size of the virus is 30,000 base pairs. And so 1% difference would be 300 base pairs. And so you somehow have to account for, if it was 1% difference, and of course this is 4% difference, 1,200 unexpected changes between these viruses. 1,200 and scattered ones, not ones that are all next to each other. And so I think that I often find that thinking about that number, it's one thing to be like, oh, so that means 4 base pairs happen to change. No, we're not talking about it's not 100 base pairs and only 4 changed. You have to be able to account for how there were random mutations at 1,200 sites compared to these other viruses that are known.
And to your point of 1,200 sites, this is not one segment of the genome that's been changed. This is all over the place.
Yeah,
So,
exactly.
So basically, and we're going to hammer on this more, no one had something close enough to SARS-CoV-2 to be able to engineer it to become a pandemic virus. This RATG13 is too distant. This 1,200 bases, by the way, it would take 20 to 30 years at minimum for a virus circulating in nature to accumulate those number of mutations, given the mutation rate of the virus.
And if you were going to engineer something in the lab to study it, for example, you would not change 1,200 bases scattered throughout the genome. That's just, nobody would even embark on such a project.
Who would even know what to change?
Exactly.
Yeah,
these are not changes that would not have any effect on proteins, and therefore you would not choose to make that kind of a decision as a scientist who's designing something, because you don't know. You would imagine that the majority of them would have impacts that you couldn't predict.
Right.
And I think that people have tried to argue at some point, oh, well, those changes came because of, you got the virus and passaged it in the lab for a while after you did all that engineering. But how do you get 1,200 changes around the virus, but none in the spots that you engineered?
Yes.
Right.
Like, it just doesn't make any sense.
All right, one more quote from point one. Quote, bat coronavirus spillover into humans is rare, end quote. I would say that pandemics are rare, but spillovers are not. Right? Before SARS-CoV-2 emerged, 4 coronaviruses made the jump. And these are the common cold coronaviruses that continue to cause seasonal respiratory disease to this day. SARS-CoV-1 in 2002 was a spillover, MERS-CoV in 2012. And during SARS-CoV-2 pandemic, we talked about on TWiV spillovers of pig and canine coronaviruses into people. Research from wildlife markets in Southeast Asia has been done to quantify zoonotic risk in these kinds of settings where you have animals being sold. And one study found that in a sampling of Laotian markets, civets, which are one of the animals sold in these markets, average 7 human contacts per hour, so that a single infected civet might get over 50 opportunities to infect a person in an 8-hour day. And furthermore, studies in rural China have shown that about 3% of the population have antibodies to SARS-related coronaviruses from bats, indicating that spillovers are not rare at all. So this is not a correct statement.
Absolutely.
Yeah.
I think that we can also think about just general how frequently are virus spillovers from animals into humans happening. Is the general idea of spillover rare? And it is very much not. It's sort of the way we think this happens in almost every other situation. So again, the idea that this doesn't happen is weird. It happens with so many viruses in so many ways.
This is a relatively recent change in our perception of how often spillovers occur, because previously we didn't have the tools to detect these unless they became pandemics.
Right.
So we said, oh, well, coronaviruses have only spilled over 4 times, 5 times, because that's the only evidence we had.
Right.
Now, in part due to not just the advance of molecular biology, but all the attention on SARS-CoV-2, now that we're looking and that we can look, we see, oh, in fact, this is a day-to-day occurrence for all kinds of viruses, coronaviruses and several others.
How would we know?
I'm sorry, Jolene.
I was going to say, how would we know that there was SARS-CoV-2 spreading in deer if we didn't have the tools we use today? And we've been talking about H5N1 in cows. And we talk about previous influenza spillovers and mixing that happens in pigs. And these are maybe things that we don't see them in our everyday life, but at the population level, they happen more than we used to think.
And since we've found out how easily SARS-CoV-2 hops species, this is an incredibly promiscuous virus. The spill back to deer, the spill back to cats, the spill back to pretty much everything with fur. I mean, we've talked about this on earlier episodes. This is a virus that can go anywhere, so it's prone to this kind of thing.
For anybody interested in a layman's read of this larger topic, I would refer them to David Quammen's book, Spillover.
Yes.
It's a great read.
Yes.
It's got lots of different examples.
I think it came out in 2013.
2012.
Yes.
So it's not, it's not as if this idea was new and just
he had no dog in this fight then.
That's right. It's actually subtitled Animal Infections and the Next Human Pandemic.
Yes.
This was inspired, among other things, by the Ebola outbreak, the big one in Africa.
Yeah. And I think there's one other piece that's sort of related to something we've been saying here about this point 1 that Dr. Chan makes. Because when I read this point 1, I feel like at least lay people are looking at it and saying, well, that can't be a coincidence that this pandemic emerged in the same city as the Virology Institute. And we've mentioned some details about how we don't always detect all virus spillovers. We, in the past, have largely detected the ones that have become pandemics. We have some pretty good evidence that we detected very early cases, but we should note that we are talking about the first cases that were detected here. And one of the things that is important in detection is sort of a good medical care infrastructure, a place where you could do that. In fact, you needed to have physicians who could say, hey, we have these patients who have pneumonia that is atypical, and we are going to actually do sequencing and find out what they have, and not just say, you got weird pneumonia, that sucks for you. If we think about a bunch of other infectious diseases we know about, we can think about what we know about their origins, but in many cases, the first ever case that was seen and published was seen in a place that had particularly good hospitals and medical care to diagnose those patients, and probably was not the first situation of an infection. I often use the first published account of HIV is a Morbidity and Mortality Weekly Report article talking about 5 patients in Los Angeles.
Yes.
But that doesn't mean that Los Angeles is where HIV emerged. It's the first place it was detected because of some good work that was done by physicians. And it should be noted that Wuhan is a city where there are these types of facilities to be able to detect these patients and diagnose them with a weird coronavirus, instead of saying, wow, that's sad that you have weird pneumonia. And so I think that some people look at this and say, well, this is no coincidence. But in fact, the reason that it is a city that has good ability to do this detection and good ability to do this care is a reason why it could be an early place where we would detect the emergence.
And also, in selecting Wuhan for this center, part of the rationale was it's a major city where we've got this kind of infrastructure, and, it's in a region where we've got a lot of people moving in and out, and yeah.
All right. Point number 2. The year before the outbreak, the Wuhan Institute, working with U.S. partners, had proposed creating viruses with SARS-CoV-2's defining feature. We're going to get back to that. All right, and it continues. Dr. Shi's group took samples from bats and other animals, as well as from sick people living near animals carrying these viruses or associated with the wildlife trade. The lab pursued risky research that resulted in viruses becoming more infectious. Coronaviruses were grown from samples from infected animals, genetically reconstructed and recombined to create new viruses unknown in nature. These viruses were passed through cells from bats, pigs, primates, and humans, and were used to infect civets and humanized mice. In essence, this process forced these viruses to adapt to new host species, and the viruses with mutations that allowed them to thrive emerged as victors. This is science fiction. This description completely overstates and misrepresents what was actually being done at the Institute. So we need to go into that so you understand that. Yang Lixi's laboratory was sampling SARS-related coronavirus from bats. This is SARS-1, not SARS-CoV-2. And the research described was based on a bat SARS-related coronavirus called WIV1, and then another version of WIV1 where its spike gene was replaced with spikes from bat coronaviruses called SCH14, WIV16, and RS4231. All 4 of these viruses are in the SARS-CoV-1 branch of the SARS-related virus family, and they're very distinct from SARS-CoV-2. Nowhere in this report is there evidence of research on SARS-CoV-2 related viruses. And so really, these experiments are relevant to the origin of SARS-CoV-2. The idea was to see if the spike proteins from these bat-related, SARS-related coronavirus could let the viruses reproduce in human cells, and if they caused a different disease in ACE2 transgenic mice. There was no forced adaptation to new species, right? Now, one of the points made in this piece is that they made more dangerous viruses, right? So one chimeric virus had enhanced viral replication relative to WIV1 in the lungs of ACE2 transgenic mice that caused greater weight loss, which is a common proxy for disease severity in mouse models. The other 2 chimeric viruses had similarly enhanced virus replication in mice, but one caused the same weight loss as the parental WIV1, and the other, WIV16, caused no weight loss at all. So there's no consistent correlation between virus replication in the lungs, and disease severity as defined by weight loss in this transgenic model. And in fact, this transgenic mouse model has no defined correlation with disease severity in humans. So all of that writing is really misrepresented what's going on there.
And this is very typical of this entire editorial, and I would assume the entire book that this writer is flogging on the world for the past couple of years, which is basically the same argument in book form, as I understand.
There's another point in number 2, which is, quote, it remains unclear whether the Wuhan Institute possessed a precursor of the pandemic virus. And I want to tell you why that is simply a wrong statement. It's very clear that they did not have a precursor. Right? There's no evidence that the Institute...
It's very clear that we don't have any evidence of a precursor, we should say.
Yes.
We can't say something's not there.
We have
can't
no
prove
evidence
a
of a
negative,
precursor. We have
which
evidence
is
that they worked with
Yeah.
SARS-CoV-1. RATG13, remember, was reported in 2016 and 2020. In 2022, a large number of sequences appeared in a database, GenBank, related to a manuscript from the WIV. And it basically provides a view into the viral sequence collection at the WIV in the run-up to the pandemic. The only viruses related to SARS-CoV-2 are RATG13 and 8 others, which are collectively called RATG15. And remember, RATG13 is 1,200 bases different. This was an unpublished manuscript in 2018, which includes phylogenetic trees confirming that no other viral sequences related to SARS-CoV-2 were being studied. Therefore, it is not unclear whether the WIV possessed a precursor to SARS-CoV-2. And furthermore, we have 2 U.S. intelligence reports that conferred this. They released 2 declassified documents on the origin of SARS-CoV-2, one in 2020, one in 2023, and both address this question, whether the WIV held either SARS-CoV-2 or a direct progenitor prior to December 2019. So the 2021 report, it's called the Updated Assessment on COVID Origin States. Early in the pandemic, the WIV identified that a new virus was responsible for the outbreak in Wuhan. It is therefore assessed that WIV researchers pivoted to COVID-19 related work to address the outbreak and characterize the virus. These activities suggest that WIV personnel were unaware of the existence of SARS-CoV-2 until the outbreak was underway. And the other report, the 2023 report on potential links between the WIV and the origin of COVID-19, reports that the lab was working on a number of coronaviruses, no known viruses that could plausibly be a progenitor of SARS-CoV-2. Information available to the intelligence community indicates that the WIV first possessed SARS-CoV-2 in late December 2019, when it was identified was from patients. And the IC, the intelligence community, assesses that this work was intended for public health needs and that the coronaviruses known to be used were too distantly related to have led to the creation of SARS-CoV-2. The IC has no information indicating that any WIV genetic engineering work involves SARS-CoV-2, a close progenitor, or a backbone that is closely enough to have been the source of the pandemic. So these are 2 U.S. intelligence reports which conclude they didn't have anything closely related.
And when they refer to the intelligence community, they're talking about the CIA, Department of Defense, people who sit in rooms perceiving China as a threat and trying to figure out what's going on there. They do other things. Right. But that's a major focus is starting with the assumption that what China is doing is going to be harmful to the U.S. In a lot of cases. And so these are not touchy-feely agencies who are trying to make everybody happy in the world. These are hard-nosed realists who perceive the entire world as a threat.
Right. And again, when I've talked to people who tried to convince me about this, and they will think that we'll have the conversation about this point, and we'll say, from our evidence does not suggest that they had this virus, that they had any viruses very close to this beforehand. What are you talking about? That it starts to become this conversation of, well, they had other ones they were getting from nature, so they just must have had some that they weren't telling us about. And it's kind of like, no, they actually have done a very good job of publishing the work that they have done throughout their careers. And so that would be a departure from everything else that they had done. They have been publishing these sequences for a long time, and there's no good reason why they would want to not tell us about things that they had in 2018 or something like that. Because for them, science works in the same way that it works for the rest of us, in that, we want to publish our work and move forward in the scientific community and show our evidence. So I point this out because in a few places where we're going with some of these arguments, you start to have to say, well, they lied, well, they faked it, well, there's this conspiracy about this. And if you really sort of add them up, we will end up with, well, they lied about like 20 things.
Yeah.
You have to sort of infer, or you have to invoke all these lies versus the way other viruses have come into the human population. And so as you're looking at these two sort of different options, realize that in some way, when you really start to poke at it, you almost have to invoke, but everybody lied all the time. To make this work, including to say, oh, well, it remains unclear, but I bet they just did and they lied.
It's definitely a very conspiratorial attitude to
Yes.
take towards it, to take towards the people who are doing the work. It's one thing to think that about a government. It's another to think that about individual scientists who are really caring and clearly over time demonstrating that they take care in how they do their experiments and how they report their experiments and data to the rest of the world.
Exactly.
All right, another point from this is we're still on number 2. In 2021, The Intercept, which is a publication, published a leaked 2018 grant proposal for a research project named DEFUSE. This was a collaboration between Equal Health Alliance, the Wuhan Institute, and Ralph Barrick at University of North Carolina. And Chan writes, the proposal described plans to create viruses strikingly similar to SARS-CoV-2. End quote. This statement is disingenuous. Strikingly similar refers only to the presence of the furin cleavage site. No, the furin cleavage site is a short amino acid sequence in the spike protein that allows it to be cleaved or cut by proteases that are present in cells. And one of the core arguments of the lab leak hypothesis is that we don't find SARS-like viruses with a furin cleavage site, and therefore it must have been added by humans, which is a crazy alternative, right? If you don't find it, you don't automatically say that people put it in. So DEFUSE was going to look at sequence data for SARS-related coronaviruses from bats, see if they have furin cleavage sites, and then if they did, they were going to introduce them into these laboratory viruses like WIV1 that we mentioned before. However, bat SARS-related coronaviruses do not have furin cleavage sites, and so they wouldn't have found one, and hence they wouldn't have introduced. This work was not funded. It's not clear that any of it was done. And so the whole idea that they were trying to create something, as she says, strikingly similar, it's just a furin cleavage site out of 30,000 bases, is simply wrong. And the closest, as we've said, the closest sequence is 1,200 bases. So you could not take that viral genome and put in a furin cleavage site and get SARS-CoV-2. One more point from number 2. This is very important. And here is her writing. One alarming detail leaked to the Wall Street Journal and confirmed by current and former U.S. government officials is that scientists on Dr. Xi's team fell ill with COVID-like symptoms in the fall of 2019. One of the scientists had been named in the DEFUSE proposal as the person in charge of virus discovery work. The scientist denied having been sick. End quote. Well, it turns out that U.S. government intelligence reports that we were citing before consistently assert that these illnesses that took place in the fall of 2019 at WIV were consistent with a variety of conditions and not diagnostic of COVID. Some were actually inconsistent with COVID-19. Now, Dr. Shi has stated publicly that none of her researchers tested positive for SARS-CoV-2. The 2023 intelligence assessment states there's no evidence that the WIV had SARS-CoV-2. And Marion Koopmans of the WHO committee investigating the origin stated there's no evidence that any of these workers had COVID. So there's zero evidence that they were sick with COVID, yet Dr. Chan says that they had COVID.
And reminder, fall of 2019.
Yes.
I was sick
Some
in
people
fall
in
of
the lab
2019.
some people in the lab came down with a respiratory virus. Gosh, what a shock.
Mm-hmm.
Yeah, it's flu season.
It's flu season. It's, yeah.
Okay. Point number 3. Shan writes, the Wuhan lab pursued this type of work under low biosafety conditions that could not have contained an airborne virus as infectious as SARS-CoV-2. Okay. End quote. Research on bat SARS-related coronaviruses at the WIV was routinely carried out at BSL-2. And the other lab that did similar work, the Barrick lab, used BSL-3. And the differences involve personal protective equipment and different engineering differences to increase containment. But the issue is not whether a lab-acquired infection could result in a pandemic, whether it did. And there's no evidence linking the WIV or any lab to the pandemic. And given there's no evidence that WIV had SARS-CoV-2 or a plausible progenitor, they could have worked without any biosafety controls at all and not sparked the pandemic caused by SARS-CoV-2. So Chan is saying that their containment would not have contained SARS-CoV-2, but they didn't have SARS-CoV-2. So all of it is moot. And finally, just note that WHO recently allows people now to work with SARS-CoV-2 under BSL-2 conditions.
And I was just looking it up. Someone can correct me if you know otherwise, but measles, which is considered to be one of the most contagious viruses that can spread between people and is similar, not a similar infection, but it is also worked with under BSL-2 conditions.
It is.
It's correct, yeah. Another quote from that point number 3, if the virus had escaped from a BSL-2 lab in 2019, the leak most likely would have gone undetected until too late, end quote.
Why? I don't even understand that point.
Yeah, because it would have, if it leaked from the WIV, that would have been the early epicenter.
Yeah.
And it's not. The Huanan market is the early epicenter.
And you would have one lineage. Sorry, but I've got to keep coming back to that.
Yes,
too.
of
That's
course.
just-
No.
You wouldn't get those amazing plots or maps from the Worobi paper that showed the epicenter so clearly as the market and even pointed out where the lab was that was not the epicenter.
One more point from number 3. Quote, in one experiment, Dr. Shi's group genetically engineered an unexpectedly deadly SARS-like virus, and she writes, not closely related to SARS-CoV-2, that exhibited a 10,000-fold increase in the quantity of virus in the lungs and brains of humanized mice. WIV scientists handled these live viruses at low biosafety levels, including BSL-2, end quote. Already discussed this above. I just want to point out that the levels of virus were determined by PCR, which does not correlate with infectivity. So 10,000-fold is not indicative of a higher virus titer. And this information is actually in year 5 progress report of an NIH grant between Ralph Barrick and EcoHealth Alliance.
Also, some of these points have been about what happens with these different viruses in mice.
Yes.
SARS-CoV-2 doesn't kill mice.
Notoriously doesn't kill mice. It's actually kind of
Well,
a
the
pain
original
for researching it.
The original SARS-CoV-2 did not infect mice. They made ACE2 transgenic mice, and the ones that are driven by a keratin promoter, they get neurological disease, which that's what kills them. So it's a completely wrong model. It's
So even if okay, let's imagine that they made this unexpectedly deadly virus in mice.
Right.
So This was a pandemic that wasn't with an unexpectedly deadly virus in mice. These things do not seem related.
Yeah,
Yeah.
they're not. This is typical of this writer and of pretty much everybody in the lab leak camp. Take a bunch of circumstantial, kind of maybe
Scary
sketchy
sounding.
sounding things and whip them up together, and they start to sound persuasive when you group them and number your arguments.
All right, point number 4, quote, the hypothesis that COVID-19 came from an animal at the Huanan seafood market in Wuhan is not supported by strong evidence, end quote. This couldn't be more false. There is so much evidence for this. Multiple independent analyses. The Warby paper is frankly devastating to the lab leak hypothesis, and that's why Dr. Chan doesn't like it. You can not like data, that's your prerogative, but she's wrong about this. Okay, so one point she makes is, quote, the bias in the search for early cases meant that cases unlinked to or located far from the market would be missed. So the idea here is, end quote. Early on, the cases were at the market, and so they're saying then people just looked around the market, and so there was an ascertainment bias. But the paper went through several advanced statistical testing to check that the results held. There is no ascertainment bias. So that's very clear. All right, another quote from this point number 4. A pair of papers published in Science made the best case for SARS-CoV-2 having emerged naturally from human-animal contact at the market by focusing on a map of the early cases and asserting that the virus had jumped from animals into humans twice. More recently, the 2 papers have been countered by other virologists who convincingly demonstrate that the available market evidence do not distinguish between a human super spreader and a natural spillover event. This, end quote. This is also wrong. There were 2 lineages of SARS-CoV-2, A and B, circulating in December at the market. There would have had to have been 2 human super spreader events to produce this pattern. In other words, someone got infected elsewhere, they went to the market and infected a lot of people. That would have had to happen twice with 2 independent lineages, which is highly unlikely.
They would have had to both get infected somewhere else and then both go to the market and both be really transmitting at the market and neither be really transmitting somewhere else.
And they both had to have acquired different strains, different lineages of the virus that are genetically distinct, which if you were working with it in the lab is preposterous.
And they went to the same part of the market.
And yes, they went to the same, right.
And super spreaders are like 10 to 20% of all infections. So it's relatively rare. So it further makes it highly unlikely that happened. But you don't even have to posit that. This paper that we've been talking about, it's Warby et al. 2022. The Huanan seafood market, wholesale market in Wuhan was the early epicenter of the COVID-19 pandemic. They consider early cases and look at their locations. And one of the best results in that paper, they test whether, so they have cases clearly linked to the market. In other words, I worked in the market and I got sick. I went shopping in the market that day and I got sick. Those are linked cases. There are other cases unlinked to the market but near it, and they wanted to know what was the difference. So they asked whether the market unlinked cases are geographically associated with the market, because that will tell you if community spread is happening anywhere other than the community around the market. So they calculate the distance from the market and where the cases were, and they show unambiguously that the market unlinked cases were significantly closer to the market than market linked. In other words, so the market linked cases, people could have lived anywhere, right? But the market linked are people in the neighborhood engaging in community activities. Maybe they went to a cafe and there was a worker from the market there, and that's how they got infected. And that didn't happen anywhere else. And these market unlinked cases are in the immediate vicinity, and it's the only place in Wuhan where this is happening. And as I said, if the spillover occurred at the WIV, you would have seen this pattern there.
Yep.
And I want to point out again, following up on Jolene's earlier comments, that we interviewed the author of this paper, Michael Warby, in TWiV 876, so you can get the story from the horse's mouth in that episode.
Yeah,
that was
a great episode.
following on this publication, right?
Yep.
Yeah.
And Chan also in number 4 writes, quote, furthermore, the existing genetic and early case data show that all known COVID-19 cases probably stem from a single introduction of SARS-CoV-2 into people, and the outbreak at the Wuhan market probably happened after the virus had already been circulating in humans.
This is
End quote.
180 degrees wrong.
This is completely wrong. She shows a phylogenetic tree in the piece, which is just a crappy phylogenetic tree. As we have said, there were 2 lineages circulating, at least 2 separate introductions. They're called A and B. They were both found in the market in December, and we know this from this environmental surveillance that the Chinese authority did. There's no question that there were more than one. And in fact, the paper, another paper in this collection, estimates that there were probably at least 8 separate spillovers from 8 infected animals to people, and only 2 of them seem to have proceeded.
Vincent, one of the things that she talks about that you've quoted is a couple of papers that are supposedly question the statistical analysis in the Warraby paper. That caught my attention. Could you address that, please?
Yeah. So one of the issues that was claimed was that there was ascertainment bias. In other words, we were looking at the market because we knew it was an early epicenter, and they used really advanced statistical methods to show that there is no ascertainment bias. And in fact, many of the early physicians were picking up the cases before they knew anything about the market. They were really good at saying, "Oh, this is an unusual respiratory disease." So that was one point. The other point, there was a mistake in the computer program that was used to calculate the probability that there was more than one spillover. So they corrected it. Someone wrote and said, "You have this mistake." They corrected it. The error gave them 99% probability that there was more than one spillover, and after the correction, it was 97% probable. So it made no material effect on the conclusions of the paper. All right, another point from number 4, quote, not a single infected animal has ever been confirmed at the market or in its supply chain. Without good evidence that the pandemic started at the seafood market, the fact that the virus emerged in Wuhan points squarely at its unique SARS-like virus laboratory, end quote. Oh my God, no one tested animals in the market. They were all taken away on January 1st and moved into the countryside or slaughtered. This is
They were slaughtered.
Dishonest statement.
They shut down the market. They killed everything. They disinfected the place.
So I want to make a statement that's sort of parallel that might help people if they're not on top of this. So the statement that Vincent quoted is, not a single infected animal has ever been confirmed at the market or in its supply chain. And I just wanted to let you know that also it has never been confirmed that my right hand was not radioactive for one day during the year where I was 17 years old. And the reason, I say that, and the reason is because we never looked. Maybe it was randomly.
Mm-hmm.
And so it's important to say, it has never been confirmed, well, because you didn't look.
Yeah.
But also note that this is something that we can't go back and check. We don't have the samples from those animals. I don't have samples from my hand when I was 17 years old. And so, yes, we are going to be able to say there's no evidence to confirm this because we didn't check at the time, and you can't go back in time and do it.
And the reason the market was shut down and everything cleared out was not some kind of cover-up. It was because there was evidence early on that the virus had emerged in the market. And so they shut down the market out of a public health concern. It made perfect sense at the time. And now, of course, we would really love it if they had maybe frozen some of those animals.
And there have been numerous historic examples of situations like this, SARS-CoV-1, for example, arising in markets under these situations. Okay, so if I were them, I would want to put a lid on this as quickly as possible.
Yeah, and in that situation, they left the market open for a while. And so those animals were still available for testing, and they did in fact find the infected animals and get that great piece of evidence there, but we can't do that here.
Which was, in the case of SARS-1, great for virology, but not so great for public health because the virus remained, the market remained open with the animals that were carrying the virus.
Yes, for 7 months because they didn't want to tell anyone that there was an outbreak. All right, finally, we're at point 5. Quote, key evidence that would be expected if the virus had emerged from the wildlife trade is still missing. Despite the intense search trained on the animal trade and people linked to the market, investigators have not reported finding any animals infected with SARS-CoV-2 that had not been infected by humans. Yet, infected animal sources and other connective pieces of evidence were found for the earlier SARS and MERS outbreaks as quickly as within a few days, despite the less advanced viral forensic technologies of 2 days ago, end quote. There has not been an intense search. The Chinese authorities are not doing it. In SARS-1, the markets were not closed. Animals were found to be infected 7 months later, not days, within days, as Dr. Chan states. MERS was identified in 2012, found in camels in 2013. So the claim of days is fabrication. And as we've already discussed, Warby et al. In a subsequent paper. So there were environmental samplings by the Chinese authorities of the market twice in early 2020. Those samples were deep sequenced and the results were posted. And the analysis of the samples show that stalls in the southwest part of the market, which is where the mammals were sold, have environmental samples that not only have SARS-CoV-2 RNA in them, but also mammal DNA from raccoon dogs and civet cats. And there's a particular stall, 6.29, which had lots of mammals. It had lots of SARS-CoV-2 positivity and had been photographed previously to have sold these animals as far back as 2019. And so we know there were, so even though we haven't tested animals directly, environmental samples show contamination with animal DNA and viral DNA. And another part of this analysis is one of the positive swabs, SARS-CoV-2 positive swabs, was from stalls with documented human cases, okay? And one of them was a shrimp vendor, who we'll get to in a moment. Stall 6.29, which had a lot of raccoon dog and civet cat DNA and SARS-CoV-2 RNA, is not associated with a known human case. All right, so we don't find human DNA in those samples. They mostly have animal DNA. And again, the animals are raccoon dogs, masked palm civets, and these are acknowledged to be source of SARS-CoV-1. Now, we can't prove this is exactly how and where the virus emerged because we can't go back and sample those animals. But this is exactly what the genetic aftermath of a zoonotic spillover event is expected to look like. And finally, let me get to Wei Guishan, who was a shrimp vendor. She got sick, one of the first known cases of COVID in Wuhan. She got sick on December 10th, and she ended up in the hospital. And she was interviewed by the Wall Street Journal in 2020 about how she thought she got infected. And looking back, there's a quote from the article. Ms. Wei thinks she may have been infected via the toilet she shared with the wild meat sellers and others on the market's west side. She said the vendors next to her on both sides got sick, and the man kitty-caught her from her almost died. One of her daughters, a niece, and the niece's husband caught the virus too. So she got sick in the market from meat sellers who in turn got sick from their animals that they were selling. It's really an interesting story. All right, we're almost done here. A couple more points. Quote, even though Wuhan is the home base of virus hunters with world-leading expertise in tracking novel SARS-like viruses, investigators have either failed to collect or report key evidence that would be expected if COVID emerged from the wildlife trade. For example, they have not determined that the earliest known cases had exposure to intermediate host animals before falling ill. No antibody evidence shows that animal traders in Wuhan are regularly exposed to SARS-like viruses as would be expected. End quote. None of these studies have been done. She writes it as if they've been done and we didn't find anything. They've not been done. So the statement is dishonest.
And the opportunity to do a lot of these studies, as Brienne mentioned, passed and was lost amid very appropriate public health measures at the time. Gee, why didn't you go out and sample a bunch of wildlife in early 2020? Well, because the entire country of China was locked down and nobody could go anywhere.
Why
Yeah.
don't we go here? I have a great experiment for you. Let's find all of the animal traders in Wuhan today we'll find all of the people today who were at that time working in Wuhan, and let's look and see if they have any antibodies to SARS-CoV-2.
Right.
It's a great experiment.
Yeah, the experiment doesn't make so much sense now, does it?
No, because they have had plenty of opportunity to be infected or to have been vaccinated.
Yeah.
So we need to know before most people in the world had those antibodies.
So Wuhan is the home base of virus hunters with world-leading expertise. Yeah, that's true. But they can't do these experiments because the opportunity to do them passed. And so that's why we don't have any of this evidence. It's gone.
All right, one more paragraph. Quote, with today's technology, scientists can detect how respiratory viruses, including SARS, MERS, and flu, circulate in animals while making repeated attempts to jump across species, which is, viruses don't make repeated attempts. Okay. Thankfully, these variants usually fail to transmit well after crossing over to a new species, tend to die off after a small number of infections. In contrast, virologists and other scientists agree that SARS-CoV-2 required little to no adaptation to spread rapidly in humans and other animals. The virus appears to have succeeded in causing a pandemic upon its only detected jump into humans. End quote.
Except for the other lineage.
Yeah.
Exactly. I mean, this is gibberish.
It's only 2 detected jumps into humans.
SARS was not detected until it first jumped into people. The first detection of MERS was in a human with pneumonia. I mean, that's how we find them when they jump into people. So there's nothing
we don't know
special.
what happened before that.
We have no idea.
This could have been going on for years.
Right.
You know, that one guy with a weird respiratory symptom who lived in a place that didn't have the ability to do really fantastic medical care and all of this virus sequencing, and so he just died of a weird respiratory infection.
Yeah.
I would just want to point out that, you know, they may say, oh, this virus was ready to cause a pandemic, but as the virus spread, it changed, right? And variants emerged that were more fit. So it wasn't really ready. I mean, that evolution during the pandemic is evidence that it was becoming better and better adapted to people. So, and the last sentence that I want to address, she writes, quote, a laboratory accident is the most parsimonious explanation of how the pandemic began, end quote. It seems to be the least parsimonious explanation, especially given all the data showing the market origin and the fact that no one had something close enough to SARS-CoV-2 to be able to engineer it. Parsimonious depends on having some information. There is none. There are no data in this piece that support a lab origin. It's just innuendo and hearsay and conspiracy.
It's only parsimonious if you believe that a whole lot of lies and cover-ups also happened.
You have to start with the assumption that the hypothesis is true, and then pick the evidence that fits it, which is exactly how this was put together.
And you, like I said, you have to keep saying, well, but that's a lie to combat other evidence, and that is not parsimonious.
No.
So if you've made it this far, folks, this opinion piece is all wrong. Just doesn't agree with the data that we've been talking about. And, you know, we have many people on TWiV who've talked about all of these data over the years. We just look at the data. We don't do any conspiracy stuff. We don't lie about it. We do the science, and it tells us that it originated in an animal. There's no question about it. Anything else?
No, it's just all too bad.
Yeah.
It is all too bad.
But, you know, the truth floats. Unfortunately, it takes a long time to get to the surface. I'm thinking about climate change and all
Yeah.
the acquaintances I have who've been climate deniers for so long, and now, it's so blatantly obvious.
I'm Vincent Racaniello. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.